Acquired immune deficiency syndrome, or AIDS, is caused by the human immunodeficiency virus. This virus targets the immune system's T cells. As you can see, there are a number of steps in the life cycle of this virus that take place once the virus infects the cell, but this research will focus on the cleavage of the polypeptides by the viral enzyme HIV protease. The inhibition of this enzyme prevents the virus from rebudding from the cell and infecting other cells. Past research has allowed for the use of compounds as inhibitors of HIV protease enzyme and are used as treatment for HIV/AIDS. However, many of these compounds are expensive and the virus often develops resistance due to the frequent use of these compounds. This makes it imperative that new inhibitory compounds are discovered through research. Oxymatrine is one such compound. The goal of my research is to test the activity of molecules similar to oxymatrine. A compound that very closely mimics the structure of oxymatrine is matrine. The hypothesis was that the ring structure of oxymatrine was what led to its inhibitory behavior. Following this hypothesis, matrine derivatives were synthesized at Meredith College and tested for inhibition using an HIV protease assay. The methods for the synthesis of matrine derivatives are shown here. Both were made using matrine. This is the methyl ester of matrine, and this is the matrinamide. We then tested all the synthesized compounds on their ability to inhibit HIV protease activity. In our test, when the HIV protease is active, a fluorescent product is created. It follows that if our test compound is an inhibitor of HIV protease, the fluorescence of the mixture is reduced. These are the results of the HIV protease assay test for the derivatives of matrine and their effects on the enzyme activity. The first bar that we see on the left is the resultant fluorescence of the control mixture containing the enzyme and substrate alone. The second bar showing the fluorescence when a known inhibitor is added to the mixture is lower than the first bar. The bars showing the fluorescence of the synthesized amide and ester are higher than the control, suggesting that the amide and the ester are activators of the enzyme. Further testing was done to check for spurious fluorescence. Various combinations of ester, amide, enzyme, and substrate were used to test if the ester or the amide have an effect on either the enzyme or substrate alone, or if the enzyme or substrate affected the fluorescence on their own. No such spurious effect was found, and it was determined that the increase in fluorescence is in fact due to the activating effect of the amide and the ester. To further confirm that the activating effect was not a result of system error, a serial dilution of the ester was prepared, added to the master mix containing the enzyme and substrate, and tested for fluorescence. The results showed that the fluorescence decreased as the concentration of the ester decreased, suggesting that the ester was in fact leading to the high fluorescence and a greater concentration of products being formed. From this research it is certain that the methyl ester of matrine and the matrinamide are activators of HIV protease. Further research can be conducted to test if the methyl ester of matrine can be used as an activator of cysteine aspartic proteases like caspases. Ester and amide derivatives of oxymatrine could also be synthesized and tested for inhibition.